Welcome to the deep dive. Today, we're looking at environments that, well, by all normal biological standards, should be completely lifeless.
Yeah, sterile zones.
Exactly. We're talking about organisms that basically treat lethal radiation, temperatures way below freezing, even boiling water. It's just another Tuesday.
Right. These are the extremophiles, and they really make us rethink what life actually needs to get by.
So our goal today, looking at the source material, it's not just saying, wow, they're tough.
No, not at all. We want to dig into the how. We're going to analyze the ingenious biochemical tricks they use. You know, the DNA repair kits, the molecular antifreeze, the protein stabilizers.
Across three big challenges, right? High radiation, deep cold, and extreme heat.
Precisely. We'll map out those specific adaptations.
And this isn't just weird science, is it? Studying these survivors gives us huge insights into resilience, but also, like, practical stuff.
Oh, absolutely. We're already borrowing their biochemical tools, things that are changing genetics, maybe even helping us protect ourselves down the line.
Okay, let's jump in. Starting with maybe the most destructive force, ionizing radiation. So let's set the scene. Ionizing radiation, we're talking UV, X-rays, radioactive decay. It shreds DNA, causes these double-strand breaks, makes reactive oxygen species. It's bad news.
Yeah, very bad. For context, just 5 gray, that's the unit of absorbed dose, 5 gray, is lethal for humans.
Okay, 5G is our limit. So who's the heavyweight champion in the radiation zone?
That title definitely belongs to Deinococcus radiodurans. It's even nicknamed Conan the bacterium.
Conan the bacterium.
Yeah.
I like it. How tough is it?
It can take an acute dose of 5,000 G.
Wait, 5,000? That's 500 times the lethal human dose.
It is 500 times, and it just shakes it off and keeps going.
That level of radiation must just pulverize its genome, right?
Yeah.
How does it even start to recover?
Oh, it does. We're talking over 80 double-strand breaks in each DNA molecule. The whole genome is basically confetti.
Confetti. Wow.
But yeah, within hours, D radiodurans puts its entire genome back together perfectly.
How on earth?
Well, a couple things. First, redundancy. It keeps maybe 4 to 10 copies of its genome handy.
Backups. Makes sense.
And second, its DNA is physically packed in this tight ring-like shape, a toroid, that keeps all the broken pieces close together.
So they don't drift far. They're already sort of pre-positioned for repair.
Clever.
Very. And the repair itself is this rapid two-step dance. Single-strand annealing, then homologous recombination. Think of it like super precise genetic cut and paste.
Okay, but here's the million-dollar question. Is that the whole story? Just good repair?
Not quite. This is where it gets really interesting. The big discovery was finding out this bacterium hoards manganese. Loads of it.
Manganese, that's an antioxidant, right, for mopping up those reactive oxygen species.
Exactly. But here's the twist. The key insight wasn't that the manganese primarily protects the DNA itself.
No. What, that?
It protects the repair enzymes, the protein machinery doing the fixing.
Wait, really? So the radiation damages the DNA, but the crucial thing is protecting the tools that fix the DNA. That seems to be the key lesson. Those repair proteins are incredibly sensitive to oxidative damage. If the manganese keeps them safe and functional, they can handle almost any level of DNA shattering.
That's kind of counterintuitive.
Yeah.
You'd think protecting the blueprint, the DNA, is job one?
That's what the research suggests, yeah. It kind of flipped the script. It implies the cell can tolerate the physical DNA, maybe sees them as manageable, but the real evolutionary pressure was on shielding the repair crew. Preventing protein damage might be even more critical than preventing DNA damage.
Okay, that's fascinating. Now let's scale up a bit from microbes to microanimals. The famous tardigrade, the water bear.
Ah, yes, the tardigrade, kind of the all-around survivalist. They can handle doses up to around 1,000 G, still huge.
And they're the ones that survive space, right?
That's them. Back in 2007, they flew some up, exposed them to the vacuum and cosmic radiation for nearly 2 weeks. Many survived.
What's their main trick against radiation? Is it just being tough?
Well, they have a general defense. They can dry themselves out into this dormant state called a ton, and hydrobiosis.
Less water means fewer free radicals from radiation.
Precisely. Takes away the solvent needed to make those damaging molecules. But they also have a specific, really unique weapon, a protein called D-soup.
D-soup, damage suppressor.
That's the one. It actually binds directly onto the chromatin, the DNA package, and acts like a physical shield.
Like body armor for the genome.
Kind of, yeah. It forms a protective coat that significantly reduces DNA damage from things like X-rays.
And the really exciting part.
The really exciting part is that when scientists put the DSUP gene into human cells in the lab...
Let me guess, the human cells became more radiation resistant.
Bingo. They showed much less DNA damage after exposure. That's a potential game changer, for radio protection. We might be able to borrow that blueprint
Yeah.
directly.
Okay, and just quickly, any larger animals show serious radiation resistance.
Well, cockroaches are famously tough, maybe 6 to 15 times more resistant than us. But the real insect champion is a parasitic wasp, Habrobricon. It can take about 180 times the human lethal dose.
180 times.
And then there's that weird finding from Chernobyl fungi growing there that seem to use melanin for radiosynthesis.
Like photosynthesis, but with radiation instead of light.
Sort of seems that way. Harnessing radiation for energy, still being researched, but fascinating.
All right, so that's handling the chaos inside the cell from radiation. Let's flip to the opposite, extreme physical destruction from the outside. Segment 2, life in the freezer.
Yeah, the challenge with extreme cold is very physical, very immediate. Your metabolism grinds to a halt, sure, but the big killer is ice.
Ice crystals forming inside cells.
Exactly. They act like tiny knives rupturing membranes. Plus, as water freezes outside the cell, it pulls water out, desiccating the inside. Remember, polar seawater is around minus 1.9 Celsius. It's already below the normal freezing point of fresh water.
So how do the microbes that love the cold, the psychrophiles, stop their membranes from just, shattering like glass?
It's all about membrane fluidity. They pack their membranes with unsaturated fatty acids.
Unlike saturated fats, like butter, which go hard in the cold.
Right. Unsaturated fats have kinks in their tails, so they can't pack together tightly. That keeps the membrane flexible, sort of oily, even at freezing temperatures.
They need that flexibility.
They do. And their enzymes, their internal proteins, are also more flexible, optimized to work fast in the cold. The downside is they fall apart pretty easily if things warm up even a bit.
A trade-off, then.
Definitely. And we see how well this works. Scientists have revived microbes from ancient permafrost, thousands of years old, basically suspended animation.
Okay, moving up the complexity ladder. Antarctic fish, the notothenoids, they swim in water that should freeze them solid.
Ah, these guys are amazing. They evolved a fantastic solution. Antifreeze glycoproteins, AFGPs.
Antifreeze proteins
Yeah.
in their blood.
Yep, circulating in their blood. These proteins latch onto any tiny ice crystals that start to form.
And stop them from growing bigger.
Exactly. They effectively cap the crystals, preventing the whole blood system from turning into slush. It lets their blood stay liquid well below its normal freezing point.
And wasn't there something weird about how that gene evolved?
Oh yeah, great story. The gene for AFGP actually evolved from a duplicate copy of a gene for a pancreatic digestive enzyme.
Wow. Evolution just repurposed a digestive enzyme gene into an antifreeze gene. That's resourceful.
Isn't it? Talk about tinkering. And then there's a subgroup, the ice fish, that took things even further.
The ones with clear blood.
That's them. They completely lost their red blood cells and hemoglobin. Their blood is milky white, almost transparent.
Hold on. No red blood cells. No hemoglobin. How do they transport oxygen? Isn't that kind of essential?
You'd think so. It seems like a terrible idea. But it works for them because that Antarctic water is so cold, it holds a huge amount of dissolved oxygen.
Ah. So they absorbed enough oxygen directly through their gills and maybe skin, just dissolved in the plasma.
That seems to be the case. It means they don't need to spend the energy making red blood cells, a very specialized adaptation for a very specific, very cold place.
Okay, and the absolute star of freezing tolerance on land has to be the wood frog, Rana sylvatica.
Oh, the wood frog. It's just biologically astonishing. This frog survives winter with 60 to 70% of its body water frozen solid.
Frozen solid, like an ice block.
Pretty much. Heart stops, breathing stops, clinically dead.
How is that even possible?
It's a masterpiece of controlled freezing. First, it uses special proteins, ice-nucleating proteins, to make sure ice starts forming outside the cells and the body fluids.
So it controls where the ice forms.
Right. As that external ice forms, it pulls water out of the cells, concentrating the inside. Then the frog floods its cells with incredible amounts of cryoprotectants, mostly glucose, basically sugar, and urea.
So the inside of the cells doesn't freeze, it becomes like syrup.
Exactly. A thick, glassy, vitrified state. It prevents ice crystals from forming inside and damaging the vital machinery. Organs are stabilized in this syrupy state.
And then in spring.
It thaws out, heart restarts, lungs inflate, and it hops away. It's like biological suspension powered by sugar.
From frozen solid to boiling hot, let's hit the other end of the thermometer, segment 3. Life at the boiling point. Cold makes things rigid. Heat makes them fall apart. Denatures proteins, makes membranes leaky. Most life checks out around 50 Celsius. Who likes it hot?
We're back in the microbial world again, mainly the archaea. These are the hyperthermophiles. We've got things like Pyrolobus fumori, which actually prefers growing at 113 degrees Celsius.
Prefers 113 C. That's above boiling.
It is. And then there's the famous strain 121, which survived being autoclaved at 130 degrees Celsius. They need fundamentally different chemistry to survive that.
Okay, starting with the membranes again. How do they stop them from just melting?
Well, instead of the ester-linked lipids we have, they use ether-linked lipids, much more stable against heat. And crucially, many of them don't have a bilayer membrane like ours.
2 layers of lipids.
Right. Instead, they often fuse those lipids into a single, rigid monolayer. It's called a tetraether monolayer.
A single layer instead of 2. Why is that better for heat?
Think of our bilayer as 2 sheets that can get floppy and leaky when hot. Their monolayer is more like one integrated, stiffer sheet. It just holds together better, prevents things from getting too fluid or leaky at those crazy temperatures.
Got it. More structural integrity. What about their DNA? Surely it's trying to unzip constantly at 100 degrees plus.
It would be, yes. But they have a unique enzyme called reverse gyrase.
Reverse gyrase, what does that do?
It actively introduces positive supercoils into the DNA helix. It basically overwinds the DNA.
Overwinds it, making it tighter.
Exactly. That makes the 2 strands much harder to pull apart, requires a lot more energy, effectively fighting off the thermal denaturation.
Clever. And proteins.
Their proteins are built differently, too. More internal ionic bonds, more hydrophobic interactions, things that help them resist unfolding. Plus, they produce huge amounts of molecular chaperones.
Like heat shock proteins.
Sort of, yeah. Things like the archaeal thermosome. These are constantly patrolling, finding proteins that are starting to unfold due to heat and refolding them correctly. Constant maintenance.
And this natural heat stability, that's where the biotech revolution came from, right? Taq polymerase?
Absolutely. Thermus aquaticus, found in hot springs around 70 degrees C, gave us Taq polymerase. Its ability to survive repeated heating cycles up to 95 C or more is what makes PCR possible.
Polymerase chain reaction. The bedrock of modern genetics, all thanks to a heat-loving microbe.
Pretty much. Its adaptations became our tools.
Now, scaling up again, what's the most heat-tolerant animal we know of?
That title probably goes to the Pompeii worm. It's a polychaete worm found living right on deep-sea hydrothermal vents, black smokers.
Right on the vents, where the water is superheated?
Yeah. It lives in this insane temperature gradient. Its tail end, which sits closer to the vent opening, can be experiencing temperatures up to 80, maybe even 105 degrees Celsius.
105 C. While its head is in much cooler water, how does it manage that difference across its own body?
It doesn't seem to be entirely about having superheat-proof proteins itself. Its survival seems heavily reliant on symbiosis. It's covered in this thick mat, like a fleece of filamentous bacteria.
A bacterial coat.
Exactly. Scientists think this bacterial fleece acts as insulation, maybe helps dissipate heat somehow, and the worm itself likely does a lot of behavioral shuttling, moving slightly back and forth to manage the exposure. It's survival through cooperation and behavior, as much as biochemistry.
Fascinating. Okay, one last example, managing heat in a totally different environment. The desert. The Saharan silver ant.
Ah, yes. This ant forages when the desert sand surface can hit 70 degrees Celsius. That's incredibly hot.
But the ant itself can't get that hot, right?
No, it has a critical thermal maximum around 53.6 degrees Celsius. It has a very short window to find food and get back before overheating.
So how does it stay cool? It's tiny, silver?
Yeah.
Is it the color?
The color is part of it, but it's more sophisticated than just being silver. It has these specialized triangular cross-section hairs covering its body. They're nanostructured.
Nanostructured hairs. Doing what?
They do 2 things brilliantly. First, they are extremely good at reflecting visible and near-infrared sunlight. That's most of the sun's energy. They bounce it away.
Okay, reflects heat.
But second, and this is really cool, they are also highly emissive in the mid-infrared part of the spectrum. That means they are incredibly efficient at radiating the ant's own body heat away from itself.
So they reflect the sun's heat coming in, and they actively dump the ant's internal heat going out.
Exactly. This dual action, reflecting sunlight and enhancing thermal radiation, can cool the ant's body by a crucial 5 to 10 degrees Celsius.
Wow. Gives it just enough extra time.
Yeah. Plus it has long legs, which helps keep its body a bit higher off the scorching sand surface. It's a combination of physical adaptations and behavior. So if you look across all of these examples, radiation, cold, heat 30, you see this common theme emerging, don't you? It's all about robust damage control.
Yeah, whether it's fixing DNA that's been blasted apart or stopping ice physically forming or constantly refolding proteins that are falling apart in the heat.
Exactly. It's redundant systems, specialized protective molecules, constant repair, mechanisms that shield the vital parts or fix them incredibly fast when they inevitably get damaged by these extreme environments.
It really shows how incredibly adaptable life can be. And like we said at the start, this isn't just, fascinating trivia.
Not at all. The practical impacts are already huge. We mentioned Taq polymerase that changed everything in genetics.
And those antifreeze proteins from fish. People are looking at them for preserving organs for transplants or even improving frozen foods.
Right. And looking forward, that tardigrade DSUP protein, the idea that we could potentially engineer human cells to be more radiation resistant using a protein from a water bear, that's straight out of science fiction, but it's being actively researched.
For things like protecting astronauts or maybe patients undergoing radiation therapy.
Potentially, yes. It's early days, but the principle has been shown to work in lab cells and even mice.
So wrapping this all up, what's the big takeaway from this deep dive into life at the extremes?
I think the biggest lesson extremophiles teach us is that our definition of habitable is probably way too narrow, too Earth-centric. What kills us instantly is just home for these organisms.
Because they found a way through biochemistry or symbiosis.
Exactly, through ingenious adaptations. It makes you wonder, doesn't it? If life has already figured out how to thrive in boiling acid or solid ice or withstand radiation, that would vaporize us.
Does that mean the universe might be way more hospitable to life in forms we can barely imagine than we usually think?
That's the provocative thought, isn't it? Maybe we should spend a little less time looking for perfect Earth twins and a bit more time considering that life might already be flourishing in places we've written off as utterly uninhabitable. Life finds a way, often in the most unexpected places.